Hello everyone and welcome to the May Soils Network of Knowledge webinar for 2016. Today I'd like to introduce Lukas van Swieten, and Lukas gets a gold star from us because this is his second SNOC appearance. He's the only one who's come back for more. Lukas is the Principal Research Scientist here at New South Wales DPI, and he's located up at Wollongbar Primary Industries Institute, as I am. As well as this, he's an adjunct professor in rural climate solutions at the University of New England and Southern Cross Plant Science. And in this capacity, he supervises numerous PhD students. He's a Churchill Fellow and a scientific panel member of the International Biochar Initiative. He's published over 60 scientific papers and book chapters with national and global collaboration. And today he's going to talk to us about a term that I'm sure many of you will have come across, soil functionality. But what does that really mean, and what does it imply for our management of soils? So thank you, Lukas.
Thank you, Abby. Now, soil functionality is a very, very broad term, and I've been asked this question. What functions do soils generally play? And the first one that we're potentially most interested in are the fact that it supports 95% of the global food production. And according to the latest FAO figures, that means that soils are feeding over 7 billion people. We have to recognise that soils support forests, which produce 165 gigatons or thereabouts of oxygen each year, and soils therefore support biodiversity and habitat. Soils also filter surface and groundwater resources, so soil and our water resources are very, very closely linked. And many of you might not realise, but it also provides an engineering medium. So, whenever we build a house or we put a road or a bridge in place, these are usually held in place by soil. So, here we can see that soils play many, many varied functions. Now, I borrowed this particular slide from the Centre for Environmental Transformation in the United States, and I hope my mouse works. Yep. We can see here that agriculture, land and soil is right in the heart of a functioning society. Now we've got farming here, which drives economic systems, which drives demand for food and population, which also has got another function of our political system. So really, without going to this slide into too much detail because we don't have time, I just wanted to stress here that again, soil is right at the heart of an active functioning society. Now, I believe it's really time for soil to perform. The fact is, and these aren't my numbers, these are from UN FAO, that the population is expected to hit 9 billion by 2050, and that's not that far away anymore. And it also means that our food production needs to double from today's numbers by 2050. So we've got some very, very significant demands on our soil. And many farmers have been trying to double their yields, and many industries are finding it difficult just to sustain or maintain existing yields, even with increased inputs. So we're going to be asking a lot of our soils over the next 30 or 40 years. Funnily enough, soils are also important for, as I mentioned before, societies. And there have been many, many papers and published reports showing that the biggest threat to global stability is food availability. And in developing countries, food availability is often the main cause for collapse of governments. And these crises are made even more important with changing climate patterns, potentially less rainfall or bigger storm surges, or indeed increasing sea level height. And what we need to stress here is that over time, certainly the number of malnourished people globally has been increasing, and at the same time our global stores of grain on an annual basis have been decreasing, and the 2013 numbers have got roughly 71 days of stored grain globally. So again, this is soil being at the heart of society, driving economies. Most of us have been to the supermarket lately and have noticed that beef is probably almost double the price of what it was a couple of years ago. And this is again an issue with increasing demand. Many countries are demanding greater levels of high quality protein, such as beef. And even in Australia, with the price going up, it means that people are probably eating less beef, or they're paying a larger portion of their salary on trying to maintain this product. So the issue of increasing prices for quality food is certainly not is not just limited to developing countries. We're seeing these repercussions globally. So this gets us back to soil. And what is soil? And here we've got some wonderful monoliths here done by Roy Laurie and Brett Enman from New South Wales DPI up in the right-hand corner. Soil is essentially around about half of its mineral material. A quarter of it is airspace or gas space, and roughly a quarter of it's soil solution. And we've got a little bit of organic matter driving a lot of these soil functions here. So again, I'm going to stress that many of the functions or the biological functions in soil are driven by this relatively what makes up a soil. So what are the key functions relating to agricultural production? Certainly the one that we probably understand most is nutrient cycling, and I'm going to talk about that in a bit more detail soon. Certainly water relations is a really important issue, and in Australia, where much of our agriculture is water limited, it plays a very important role. Soil is, of course, as I mentioned before, pivotal in driving biodiversity and habitat for both above ground and below ground organisms. It filters and buffers, so it's important for keeping waste, for example, heavy metals from waste from leaching into groundwater resources. And we have to remember that the majority of our wastes are put back onto soil. So it's something that we need to recognise that not only is it used for agricultural production, it's also used as a waste dump. And certainly physical issues. So soil can also be used there just to hold plants up. Certainly the structure of soil is important for root development. Root development is important for maintaining a crop upright and stopping it from lodging. So there are many functions here that soil is playing. One of the important aspects here is that just looking at the grains industry, and this information is from a web page called Yield Gap Australia. So this is APSIM modelling, looking at the potential yield and the actual yield of grains in Australia. And this takes into consideration climate and water. So it takes out a couple of those limiting factors. And even with those limiting factors, those two key limiting factors taken out, I'll just skip straight to this right-hand column here. Basically, across Australia, we're lucky to get 50% of the maximum yield that a particular soil under a given climate and rainfall scenario is capable of delivering. This gap in yield is essentially due to soil function, and probably most likely due to soil chemical and biological functionality. And I'd just like to step through a few of these points now. So what are some of the driving forces affecting soil function? And certainly it's clear that soil function is a collaboration between the plants, the soil microbes, and the inherent mineralogy, given to a particular agro-ecosystem. So, for example, different soil types in different regions are going to allow different functions to occur. And certainly the carbon supply and the carbon cycle is a motor that essentially gets all of these cogs turning with nutrient cycling. And there's so many aspects to the carbon cycling that I certainly can't won't go through them all today, but I'd like to just give a few examples now. So here we've got a very simplified version of nitrogen cycling. So we've got a nitrogen fertilizer going onto soil. So that fertilizer could be in the form of urea, it could be in the form of fixed nitrogen from a legume, or it could be a manure, or even a natural nitrogen coming in from the atmosphere. It enters the soil and ultimately converts through mineralization into ammonium, depending on the soil pH. During this cycling process, it nitrifies through to nitrate. Nitrate then has got a couple of major loss pathways, so it can leach through the soil profile, but it can also then denitrify. So this is also a part of the denitrification cycle, converting to nitrous oxide and dinitrogen. How do we quantify this, and why do we want to know these particular factors? So there are lots of ways to look at this particular cycle, and some of the really high-tech, expensive methods are based on metagenomics, gene quantification, DNA profiling. The problem with some of these methodologies, we're looking at these specific genes here, for example, that are responsible for the different pathways. It's an expensive process, it's time-consuming, and you're using a very small amount of soil, and it's an absolute snapshot of what's happening at a particular point in time. It doesn't tell you what's happened previously, and it's not going to tell you what might happen tomorrow. So what you're essentially looking here is what genes are present, and it really doesn't even relate to how active these genes might be. Another way you might be able to look at these particular soil functions is a technique known as microrespiration or community structure substrate utilization. It uses a larger volume of soil, and essentially you can put a substrate into that particular soil and you can see how it converts to another form through breakdown of carbon substrates and then capture of these carbon substrates. You can look at enzymes. And the beauty with the enzyme technique is you're using a larger volume of soil, and essentially you're looking now at the potential for soil to do a particular function. Or you can do a soil test, and this is probably the thing that most soil scientists would be doing on a regular basis. It's a much larger volume of soil, and you're looking for the key things that the plant's interested in. So for example here, the ammonium and the nitrate. The point I want to make here is that each of these particular ways to look at this particular soil function, they've got a different purpose, and the purpose of for research, trying to unravel particular mechanisms, or what an agronomist might be interested in with regards to crop nutrition, will require a different way to look at this particular cycle. So I just wanted to point this out as a particular example of nutrient cycling. So here's an example of the team. So credit Mick Rose for this particular work. So this is a part of our GRDC project looking at the impacts of herbicides on soil functionality. So here we've got a chromosol which has been incubated at moderately high water holding capacity, and in that particular soil we applied herbicides including glyphosate or Roundup and diflufenican. So the first one is glyphosate at a label rate, and then glyphosate at 5 times label rate application. Diflufenican, same deal, and tibuconazole as a fungicide as a bit of a positive control. And here we're just looking at nitrate concentration as I guess an ultimate indicator of the nitrification cycle. Here we can see the control has generated at this point in time about 175 milligrams of nitrate in that particular soil. Glyphosate really hasn't had any impact whatsoever on the nitrification cycle. However, here we look at diflufenican, and we've had a statistically significant inhibition of the nitrification cycle with that particular herbicide. Interestingly, with the fungicide at label rate application or at low rate application, there was no impact on nitrification, but if we applied a slightly higher rate, we had an impact on nitrification. So again, this is just an example of where a stress factor might impact a particular cycle in soil. Now, I also want to point out here that just because we've got nitrification being inhibited, it doesn't necessarily mean it's a bad thing. Remembering that there are many fertilizers now that have got nitrification inhibitors in them, and that's to stop the production or to lower the production of nitrate in soil and therefore substrate for nitrous oxide or nitrate leaching. So we just need to take this into consideration, that this particular example of suppressed nitrification is not necessarily a bad thing. And I'd just like to show some data from Enzyme's experiment in Wagga. So this is just looking at some enzyme activities after a very common practice of liming. So most farmers would add lime to soil to fix up a pH constraint, such as aluminium toxicity. But the role of lime goes a lot deeper than just simply changing a soil chemical attribute. So obviously, the farmer is most interested in changing the pH for that soil chemical constraint. However, other things happen. So we're looking at some enzyme activities in this particular soil. So we've added, SR's added lime, so no lime, lime at 4 tonnes per hectare, and lime at 10 tonnes per hectare. And we've added this at different layers in the soil. So the surface soil at 10 to 20 centimetres, and soil at 20 to 30 centimetres. And this is a part of SR's soil subsoil acidity program. And we've had a look at the impact of lime on carbon cycling, and here we're using an enzyme called beta-glucosidase. Then in this particular instance, lime has got no significant effect on carbon cycling. However, we have a look at nitrogen cycling, in this case aminopeptidase, and what we're showing is a very consistent trend of very significant increases in aminopeptidase activity with lime addition. So one of the possible reasons for this, and obviously it needs a lot more research here, is that when you're increasing the pH of soil, you're now allowing some of the proteins, which are mildly positively charged, to release from the soil clay particles. So now these proteins or proteinaceous materials are becoming available in soil solution at more neutral pH, which means that there's more substrate now available for microorganisms. These microbes are seeing this substrate availability and they're going, yum, we're going to now turn these proteins into into food for us. And that's essentially an outcome of the aminopeptidase enzyme. Interestingly, when we look at a phosphorus enzyme, we're getting pretty much a significant decrease between the 4 tonnes of lime through to the 10 tonnes of lime per hectare. This is, we're finding it difficult to explain this result. It's a little bit against what our hypotheses were at this particular point in time. Again, when you neutralise or increase the pH of an acidic soil, one would expect that there is more phosphorus availability in that particular soil. Therefore, we were expecting an increase in the phosphomonorespiration enzyme. But because there's more chemical phosphorus available in soil, the microbes have to work less hard to actually access phosphorus for phosphorus cycling. So, again, we certainly don't have a very strong handle on the exact mechanisms for this, but we are seeing this particular outcome from liming, and obviously there's a lot more work to do here. An example of enzymes being used to unravel some pretty basic soil functions. Here I've got some work from Dr. Flavio Fornazio, who was here a couple of years ago, and he was looking at enzyme activities and how it related to truffle production, for example. And essentially what he was finding is in areas of the paddock, so each dot here represents a tree, and he was finding high truffle contents where there was high alkaline phosphatase, and very low truffle content where there was grass cover and higher beta-glucosidase enzymes. So it's a bit hard to actually determine which factor is the main cause here for truffle production, but certainly the enzymes were useful in helping to diagnose some of the areas or potentials for these soils to produce truffles. Clearly, soils provide a very significant biodiversity and habitat. And here's an example of an avocado orchard. So this is fairly old work, but I thought it was quite pertinent to this particular seminar. One of our collaborators has got an avocado orchard, and you can see here a very distinct layer between the A horizon and the organic horizon. So we have about 10 centimetres or so of organic material on top of the soil. Some would say, oh, lots of organic matter, that's a great thing. However, what we're not seeing is a term called biotubation, and that is essentially driven by soil animals, things like earthworms and ants. And earthworms are certainly an important aspect of a healthy, functioning, biodiverse soil. Essentially, what we diagnosed this issue down to was the fact that this particular soil has had 30 or 40 years of copper application as a fungicide, and essentially this copper now is quite toxic to earthworms, and fairly simple OECD tests known as the earthworm avoidance test, where we have our contaminated soil and a non-contaminated soil, and we put earthworms in the middle, and we come back 3 days later and count where they are. Basically, all of these earthworms migrate to the non-contaminated soil. And here we can see where we've done a bit of science, the threshold seems to be around about 50 parts per million. So if you've got copper in your soil at about 50 parts per million or over, they're the kinds of concentrations where you're going to start getting significant avoidance or by earthworms. So again, here we've got earthworms as an indicator of biodiversity and soil functionality. And of course, I guess in today's climate, one of the important aspects for soil functionality is its ability to sequester carbon. And here we've got a picture of a global carbon cycle and basically where carbon sits. And this little thin layer on top of the Earth's crust is known as the soil, which we all know, and it's a really important store for soil carbon. Again, the soil is supporting the forests and the agriculture, which are driving photosynthesis. And the photosynthesis is what's responsible for grabbing carbon dioxide out of the atmosphere, turning it into sequestered forms of carbon, as in wood or organic matter in soil, and releasing oxygen back into the environment. So again, here I just wanted to stress the importance of soil both as a store for carbon, but also as a medium allowing these particular carbon cycles to be driven through natural processes. And here's just an example of some of the trials at Wollongbar, where we've been having a look at, I guess we'll call it, intercepting the carbon cycle. So can we use materials such as pyrolyzed biomass, or better known as biochar, for intercepting the global carbon cycle? And some of the work here from PhD student Han Weng has shown that we've got very significant opportunities here for storage of new carbon. So not only from the carbon added from the biochar, but this carbon now is inducing a process called negative priming, whereby it's stabilizing new carbon inputs from photosynthesis. Look, that's been my 20 minutes. I've covered a lot of topics very, very broadly, I guess. And Abby asked me just to give a summary slide at the end, which I've done here, and I've just circled the organic fraction of soil. Again, this is a motor driving much of the soil functionality. It's something that we don't understand as well as we should, and I think this is an area where if we're going to double our food production within the next 30 years, we're going to need to exploit soil functionality for this. We can certainly add increased inputs of fertilizer into soil, but we know that that doesn't always lead to the desired outcome that we're after. What we need to do is drive this soil functionality to get the yield increases for the increasing population. Thank you.
Thank you very much, Lucas. That was fantastic, and I think you raised some really interesting and important points. I had one question around the terminology of soil functionality, and obviously our interest here is how that relates to food production. But does that the definition of it change, or what's important change depending on how you're looking at using the soil, Lucas? So if you weren't looking at it from a food production perspective.
Sure. Look, some of your questionnaire actually dropped out, but I'm gathering it was basically about do inherent soil conditions or the location of the soil in an agro-environmental climate dictate what the potential of the soil is, and the answer to that is absolutely yes. So, for example, if you're looking at a function of a tennis hole in Western Australia, where they've got 300 millimetres of rainfall, your absolute food production in that particular situation is going to be much, much lower compared to a vertisol on the Darling Downs with a rainfall of 1,200 millimetres per year. So, we certainly need to understand that different soils are going to function very differently. What we need to try and understand is mechanisms to optimise a particular set of functions for that particular outcome. So, for example, soil will function differently for agricultural production compared to producing a forest, for example. We know that there are very, very different in agricultural soils compared to a more lignin-cellulose, more lignin-based forest scenario where you've got very, very different carbon inputs into soil and different qualities of leaf litter and organic matter being returned to soil. So yes, functions are very different, and they're dependent upon what you're asking from the soil at that particular in that particular season.
Yeah, and I think that's a good point to make. Thank you. I have a question here from Eddie Joshua, and he said, thank you for the talk, but he also wants to know is, can we influence water content to aid yield increase? Would you like to comment on that, Lucas?
Yes, I believe water content in soil, certainly water holding capacity, not so much water availability, but certainly water holding capacity can be influenced through soil microbiology and optimizing soil functionality. We know that increasing organic carbon content can increase soil's ability to hold water. There are also opportunities to improve water and gas flows within soil. So, for example, a compacted soil will have a lot more surface flow than a soil that's not compacted, or a soil with good airspace and good pore structure. And again, microbiology and agronomy, so careful selection of plants and building up that soil structure can certainly influence the water relations in soil. There's no magic formula though.
There's a few questions about the WOGA information that you presented. Different people wanted to know whether it was surface applied or incorporated, and what the pH of that soil was. I'm assuming it was before the lime was applied.
Sure. So each layer was amended individually, and it was incorporated evenly throughout that particular soil profile. So these were incubation tests where, so layer was removed, the 10 to 20 centimetre layer was removed, and the 20 to 30 centimetre layer was removed and treated as an individual, soil profile, where the lime was mixed evenly throughout that particular profile. And that's a part of Hassan's project, where he's looking at mechanisms to get lime, for example, deeper into the soil profile to alleviate some subsoil acidity constraints. I understand that soil had an initial pH of around about 4.7 in calcium chloride. I can certainly find out, but that's what I understand. and it certainly had a slightly more acidic subsoil.
And I'd just add to that for people who are listening who are unaware, Esine is a researcher here at New South Wales DPI who's located at Wagga. So, if you would like to get more information about what he's doing or that particular project, you could contact me and I could put you in contact with him. So that's the addition for that bit. Greg Chapman's also said thank you, and he's got a question here. Would you expect addressing the most limiting factor for production also maximizes soil microbial activity? Are there any studies which show this?
Yeah, so more microbial activity is not necessarily what you're trying to achieve. So again, every single soil, for every single crop is going to have a different limiting soil function, and it might not be related necessarily to soil biology or soil microbial activity. But certainly what's most important, I think, is to understand the particular functions of that soil and not performing optimally for that particular crop or situation that we're asking from the soil. Again, a lot more work has to be done to actually understand, and I think I've stressed this many times before, what the key functions are in soil. It might be related, as I said before, to water, structure, nutrient availability, nutrient cycling, carbon cycling. Certainly improving particular functions through application of organic matter or application of nutrients or lime are things that are currently being researched globally. But again, these are going to be very different for every soil.
Thank you. And I guess a further question, which is a bit in that vein from Stephanie, she said, is providing more food for a soil organism to the soil system going to automatically lead to more nutrient cycling in the organic nutrient pools? And if so, how long would you expect that to happen?
Yeah, look, BP probably be the best person to answer this question. But essentially, if you start driving that soil carbon cycle, which is based on how much food is available to the microorganisms in the soil, you now start driving nutrient availability. So look, the two of them are very, very earbox. You start driving one particular gear, which is a carbon cycle, which is central to everything. You start driving that carbon cycle in soil, you then start driving everything else that's happening in soil as well. So that really is the heart of soil functionality, and Stephanie really hit the nail on the head there.
I'm just going to unmute BP. He is actually online today. And would you like to comment further on Stephanie's question?
Okay, there are a couple of comments I wanted to make. First is about the last slide, the importance of organics. I feel like it's really kind of important. We're doing lots of research on soil organic matter, and we're just working on, we're trying to understand organic and linking it with productivity. So we really need to understand that. I mean, it's really important. But the problem is in Australia conditions, the soil organic matter content is very low. So we really need to enhance our ability, how we can increase carbon, what management practices is going to increase carbon. And carbon, for example, allocation in the system. So you have photosynthesis enhanced, and if photosynthesis process is kind of enhanced and carbon allocation enhanced, you have a very close linkage between carbon and nitrogen. And obviously, this is all going to contribute to soil functionality, such as microbiology, those kind of things. And really important, we really need to understand the link between carbon and nitrogen cycle, as Lukas mentioned in one of his slides. And I was going to actually ask or make comments on these questions, but good that this question came through. So we're working on linking carbon and nitrogen cycling, and an allocation in the system, you're going to have more nitrogen use efficiency. So there could be more nitrogen going into the grain, so you improve the quality of the grain and food quality, etc. And probably as well.
Thanks, BP. And for those of you listening who don't know, BP is also a researcher here at New South Wales DPI, and currently doing some research on soil functionality and carbon and things like that. So thank you very much for those comments, BP. There's one other question, Lucas, on my list here from Harry Kibler, and Harry has asked, as a horticulturist, what's the biggest step that we can do to increase soil functionality?
To increase, look, I think again at the heart of soil function is organic material and carbon, and if we can drive increased carbon cycling in soil through increased carbon allocation in soil, as BP mentioned before, that's going to be the first step to improving nutrient availability, nitrogen availability in soil. What we don't understand that well yet is how, for example, carbon cycling in soil will buffer soil against disease, will improve uptake of other nutrients, will improve root penetration of soil, and what we're trying to achieve in soil is roots that are adventurous, and they're going to explore lots of different parts of the soil profile. And if we can maximise that, that gives us the greatest opportunity to then maximise productivity through, as I said, yield improvements, through lower risk of disease, through improved food quality as well.
Thank you, Lucas. I guess, would I be right in saying it's a bit like an engine? So you're trying to fine tune the engine so that you get the best performance out of the engine without it chewing up a lot of fuel and things like that. So it's not necessarily that you want everything to be more, it's just that you want it to work more efficiently and effectively for what you want. Would that be right?
Yep, very eloquent.
Okay, thank you. Thank you again, Lucas, and thank you everyone for attending today. See you next month. Bye.